Hello, this is Dr. Preeti Avari. So today we are starting with the third lecture in microscopic anatomy, that is microscopic structure of muscle. Now what is muscle? Muscle is a specialized tissue. Now what is the property of muscle? It can shorten in length, it can contract. Now various parts of a muscle cell are given various names like muscle fiber or muscle cell or myocyte are one and the same. The cell membrane of a muscle is called as sarcolemma. The cytoplasm is called as sarcoplasm. Endoplasmic reticulum of a muscle is called as sarcoplasmic reticulum. As you know, there are three types of muscle: skeletal muscle, smooth muscle, cardiac muscle. Now you know the location of these muscles. Skeletal muscles are attached to the bone, like bones of the upper extremity, bones of the lower extremity, bones attached to the rib cage, and the abdominal muscle. Smooth muscles, these are present in the wall of viscera, like wall of gastrointestinal tract, the wall of ureter, urinary bladder, uterus, vas deferens, like that, and also in the wall of blood vessel, and a muscle present in the skin called as erector pylorum, and muscles of iris, sphincter pupillae, dilator pupillae. And as the name suggests, cardiac muscle will be present in the wall of the heart. This is a musculature of the heart called as trabeculae carni. Let us begin with histology of skeletal muscle. Skeletal muscle is a voluntary muscle innervated by somatic nerves. That means, for example, if the muscle is present in the upper extremity, it will be innervated by some branch or other branch from brachial plexus. Now, how these muscles will look like if we see them under low power. The muscle fibers are elongated and they are cylindrical. They are placed one over the other. So, as if one cylinder is placed on the other cylinder, or the big cells which used to come previously to charge, which are present inside a torch, those cells, as if you are stacking them one over the other. So, this way, the cylindrical muscle fibers will be placed one over the other. Their length will vary depending on their location and their function. Some are hardly few millimeter long, and some will be going till approximately 35 centimeter, like a muscle of the lower extremity, anterior compartment of thigh called as sartorius. It is approximately 35 centimeter long, and the diameter will vary from 10 to 100 micrometers. The myocytes will be arranged in bundle, or they may be flattened as if in the muscles of anterior abdominal wall. The muscle is flat, but still the myocytes are arranged in bundle. The fibers will run parallel to each other. Fiber means the myocytes. Muscle fiber will run parallel to each other. The sarcoplasm is filled with longitudinal fibers called as myofibrils. The whole of the cytoplasm of a myocyte will be filled with myofibrils. What are these myofibrils made up of? They are made up of myofilaments, actin and myosin filaments. Now, in the skeletal muscle, we can see cross striations because of regular arrangement of these myofilaments, actin and myosin. So we can see dark band, light band, dark band, line band. This is because of some kind of regular arrangement of these myofilaments. The name striated muscle is given because of the striation seen as dark and light band on hematoxylin and eosin stain. Now we saw the shape of a muscle. What about inside? Inside, there will be multiple nuclei present in a single muscle fiber, and the nuclei will be cigar shaped, quite big cigar shaped nuclei. These nuclei don't have place inside in the center, as the center is completely filled with myofibrils. So where will they go? They are placed at the periphery. So all the nuclei are present at the periphery. And if you see the fibers will never branch. You can hardly see any junction between the fibers, cell junctions between the fibers. Here the fibers will not branch. So if you see the slide actually under low power, this is how it will appear. Now the first picture is longitudinal section of a skeletal muscle. So you can see these skeletal muscles. They are parallel to each other. They are cylindrical in shape. You can see the cross striations which will stain with eosin. So these cross striations are formed because of regular arrangement of actin and myosin. Now, where are the nuclei? The purple color dots which are present here, these are cigar-shaped nuclei present at the periphery. Now, in between, even in between the muscle fibers, if you see, there is a space. In this space, you can see that the muscle will be covered by connective tissue sheath that is called as endomysium. Now, while taking photograph, the endomysium hasn't come clearly, but endomysium will be also stained with eosin. That's a connective tissue sheath. So myocyte is covered by endomysium. Each and every myocyte will be covered by endomysium. Now, if you see the transverse section of a skeletal muscle, how will it appear? In the transverse section, you can see big polygonal sections. All sections are of same size, and there will be nuclei present at the periphery. So it's not like that you will see only single nucleus, you can see multiple nuclei at the periphery. And in the center, there will be myofibril. Center will be completely filled with myofibrils. Now, in between what you can see this eosinophilic lines, what is this representing? This is representing the epimysium covering each myocyte. So, in between the fibrocytes, their nuclei will be present, blood capillaries will also be present. Now we'll talk about the coverings of a muscle. The muscle is covered by epimysium. The whole muscle will be covered by epimysium. The bundles of muscle fibers will be covered by perimysium, and a single myocyte will be covered by endomysium. What are the functions of these coverings? They will carry lymphatics, blood vessels, and nerves to the muscle. They will provide shape to the muscle, and they help in transmission of force of contraction of a muscle. Now we'll come to the electron microscopic structure of a skeletal muscle. If you see it under electron microscope, you can see the basic structural and functional unit of a muscle called as sarcomere. What is this? This is a part of a myofibril. We talked about myofibril. Myofibril are going to completely fill the muscle fiber. There are many myofibrils are present in a single myocyte. So within the myofibril, between two Z lines, the structure which is present is called as sarcomere. Dark and light bands of all the sarcomeres of parallel microfibrils will coincide with each other in a muscle. Of all the myofibrils, all the sarcomere, their dark and light bands are going to coincide with each other. And we can see some kind of dark and light bands and lines inside under H&E staining in the sarcomere. So we will be talking about them. What is A for, what is I for, what is Z for, what is M for, what is H for. A is for anisotropic, I for isotropic, Z is Schwiesen-Schiebe, M is Mittelschwebe, that is in the midline, H is the line of Henle or Henson's band. Let us try to see it. Now the first picture is of a relaxed sarcomere and the second is of contracted sarcomere. Now this sarcomere is a part of a myofibril. So this is a small part of a myofibril between two Z line. So these are two Z line on periphery. To the Z line attached, the shown with black color, are actin filaments. These are the actin filaments attached to the Z line on both sides. Now, actin filaments are not reaching till the midline of a sarcomere. There is a gap between two actin filaments. Now, in between the actin filaments, parallel to them, will be placed myosin filaments. So, these are the myosin filaments shown with the brown color. They are present in the midline. At some regions, you can see actin and myosin filaments are overlapping each other. Now, we will talk about the lines. We will start from the center. The center of the myosin filaments. There you can see a line that is called as M band, Mittelschwebe, in the center. After that, you can see the actin and myosin filaments. They are overlapping each other. That is going to form a band. That will be an isotropic band. After that, you can see at the periphery only actin filaments are present nearer to the Z line. That gives a faint appearance in H&E stain. We call it as I band. So this will be the I band. And at the periphery there is a Z band. So in the center there is a M band, then comes A band, and then comes I band. And after that you can see there are Z lines. So this is a relaxed sarcomere. Now we talked about the microscopic structure of the skeletal muscle. Now functionally we can divide skeletal muscle into type 1 and type 2. So what are these? The type 1 fibers are slow twitch and type 2 fibers are fast twitch. Type 1 fibers are required where you want to stand for longer period of time. So they will maintain the posture. So if you want to stand for a longer period of time, the muscle should contract for a longer period. So there will be more amount of myoglobin and cytoplasm in type 1 fibers. More number of mitochondria will be present in them and there will be rich blood supply. Type 1 fiber contraction will be slow than type 2 fiber. The type 1 fibers will fatigue slowly. They can't fatigue as easily compared to the type 2 fiber. Example of type 1 muscle is soleus and example of type 2 fiber is gastrocnemius. We are done with skeletal muscle. Now we will move on to microscopic structure of cardiac muscle. Cardiac muscles are also striated muscles. That means there will be a regular arrangement of actin and myosin filaments as we saw in the skeletal muscle. But these muscles are not under our control. They are involuntary. They will be innervated by autonomic nervous system, sympathetic and parasympathetic. Here, the myocytes will be comparatively shorter than that of the skeletal muscle. The length is varying from 50 to 100 micrometer and width is 15 micrometer. The specific feature of these myocytes are they are cylindrical, but the myocytes will branch and they will anastomose. They will join with each other. They branch, they join, they branch, they join. So they are forming a functional syncytium. So that they can contract together. We call it as functional syncytium. Now at the junctions where the myocytes come in contact with each other, against the eye band, intercalated disc can be seen. But it's very difficult to see this disc under low power. It appears zigzag in fashion in high magnification. The nucleus of a myocyte will be quite big compared to the cell size. It is oval, it is single, and it is present in the center. Around the nucleus, we can see cell organelles. Because of that, if you see the cell under H&E stain, around the nucleus, we might see a central hollow. A central transparent appearance can be seen because at the center, many cell organelles are present nearer to the nucleus. Few cardiac myocytes are modified to form conducting system of the heart. So this is the longitudinal section of a cardiac muscle. What you can see, the cylindrical muscle fibers. These cylindrical muscle fibers has got a single nucleus at the center. Cross striations can be seen. You can see the muscle fibers are branching and they are anastomosing, branching and anastomosing. So they are forming a functional syncytium. Now here we have zoomed out and we are drawing a simple diagram, but you can't see too many gaps like this in between the muscle fibers. They're very close to each other. If you see the picture, it is totally different when you see under slide in the slide. So it's very difficult to identify the slide of cardiac muscle. You need to develop your eyes for that. So you need to see them again and again. Histology of smooth muscle. In the longitudinal section, the myocytes are spindle shaped or fusiform or pear shaped. They are elongated. So in this diagram, you can see the pear shaped myocyte. You can see a bulge in the center and the ends are tapering. Length can vary from 30 to 300 micrometer. Now in the blood vessel, they are very small compared to the pregnant uterus. Here, the myocytes are held together by external lamina so that they can contract together. The nucleus will be present in the center as shown in the picture, and it is single and oval nucleus in the center. So this kind of arrangement of smooth muscle where the smooth muscle fibers are running, you can say they are parallel to each other, but what is happening is at the tapering end of one cell, the bulge of other cell is present. So at the tapering end of one cell, the bulge of other cell will be present. This is how they are arranged. And they are forming muscularis externa, muscularis mucosa in most of the hollow viscera in GIT and also in ureter. They are also present in urinary bladder in the vas deferens, in urethra. Now, why it appears smooth? Why can't we see striation? It is a muscle, so actin, myosin filaments will be present inside, but they are not regularly arranged. That gives a smooth appearance to this muscle. These muscles are involuntary. They are innervated by sympathetic and parasympathetic nerve fibers. Gap junctions are present between myocytes, which help in simultaneous contraction. Location present in the wall of hollow viscera as GIT blood vessels, in the tunica media in the blood vessel, in the ureter in its wall, in the wall of fallopian tube, in the wall of vas deferens, in the wall of uterus, vas deferens, in the wall of urinary bladder. And separately, if you want to see them, in the skin, erector pylori muscle, ciliary muscle, sphincter pupillae, dilator pupillae in the iris. Now, you need to differentiate between the transverse section of the skeletal muscle and smooth muscles. How can you differentiate? In the smooth muscle, if you see the transverse section of a muscle, all sections will be small and rounded. Some will be very small if the section is passing through the tapering end. If the section is passing through the central bulge, comparatively it will be bigger. But all the sections will be rounded with variable sizes. If the section is passing through the center, then only you can see nucleus in the section. Otherwise, you can't see nucleus in the section. You can also see covered by lamina, external lamina. These muscle fibers are covered by external lamina. If you want to compare the microscopic structure, how can you compare? The shape of cardiac and smooth muscles cylindrical, but cardiac muscles are smaller in size. The smooth muscles will be, sorry, the skeletal muscle and cardiac muscle, they are cylindrical, but cardiac muscles are small in size. The smooth muscles are fusiform or spindle shape or uh, pyriform shape. Cross striations are present in cardiac muscle and skeletal muscle. They are not seen in smooth muscle. Single nucleus is present in both cardiac muscle and smooth muscle. Multiple nuclei at the periphery present in the skeletal muscle. Only in the cardiac muscle we can see branching of the muscle fiber. Intercalated disc is seen in cardiac muscle. No connection between the cells in skeletal muscle. Gap junctions are present in the smooth muscle. Skeletal muscles are innervated by motor nerves and sensory nerves, that is somatic nerves. Smooth muscle and cardiac muscle are innervated by autonomic nervous system. Skeletal muscles are attached to the skeleton, can be seen in the upper esophagus, in the tongue. You can see in the eyelid, in the lip. Smooth muscle present in the hollow viscera, blood vessel wall, iris, erector pylorium. Cardiac muscle is forming the musculature of the wall of the heart. Thank you for watching this video. Thank you.